Okay, so I think I spent some time on some of the subsets of T cell because one of the things that you may have noticed was that T cell sees different antigen. We talked about extracellular bacteria, we talked about intracellular bacteria, viruses, fungi, so on and so forth. So each of them presents a different challenge to a T cell. And since it's an adaptive immune response, so we take them differently. And the way the T cell does it, it creates subsets. And it has an effector response, immune response. And I told you that there are many different types of effector responses. One of the effector responses is that T cell proliferates. That is important. Number two is that T cell produces cytokines. Number three is that T cell divides into different subsets, right? And we've been talking about some of the important subsets like Th1, Th2, Th17, right? And this was the last one that I thought I'll spend some time because that's going to be the basis for the clinical immunology part. We're going to start that tomorrow, and I'll begin with the cytokines again in detail and take autoimmunity. So if you want to understand autoimmunity, you can see foreign and self-antigens are recognized differently by our T cells. Now, having said that, that's not the end of the story, that's rather the beginning of the story. And one of the most important cytokine that is required for that is TGF-beta. That's a transforming growth factor. TGF-beta is the most important. So what happens is that if you have TGF-beta in an area, it's going to act on a T cell, which is of course a CD4 T cell, and this CD4 T cell will use the signaling pathway that we've been talking over and over again. But the most important player, and I've seen your textbook mentions about the most important transcription factor. So whatever you see in the belly of the T cell over here is one of the transcription factor. So the transcription factor for this particular response is FOXP3. And then this FOXP3 will signal for the T cells to produce a very specific kind of a subset of a T cell, which we've been labeling as Tregs, regulatory T cells. Another thing you would notice is that there's a small I over here, and this is the term that we normally use if you were to say that this particular cell is induced as compared to naturally occurring. If it is naturally occurring, we don't put anything over there. We just say Tregs. So what we assume is the Tregs which are naturally present. But since these are induced by these antigens, so we call them inducible or induced Tregs. Now this Tregs again will produce TGF-beta. I remember, I would say, like when in first year medical school, when we were taught about the Tregs, they used to call them suppressor T cells at that time. So they used to think that we have Th1 type cell, Th2 type cell, and this like a referee or umpire is going to make sure which way the immune system is going to cave towards. So we used to call them suppressor T cell. Then they went out of the fashion, and then it came that they were not basically suppressors per se, but they are regulators. So the same cells in the olden books, if you want to see, they call suppressor T cell, but now they've been called Tregs. And you can see the response still says suppresses. So they use the effector function as suppresses because that was not the only function they had. If I was to say suppressor T cell, you would think they would just suppress. So they still maintain the responses which are mostly suppression of all these cells, but by the end of the day, suppression would mean, in immunological term, immunosuppression. That's not good. You just want to control, you want to regulate. So we call them Tregs. Okay? Right. So the other concept I think I mentioned was, because I usually gather that by the time it's 10 to the hour, you all starts wrapping up, and I see a lot of commotion, and whatever slides I show in the last 5 minutes, probably they're just passing over your head. So this was an important concept, and I'm going to ask you, because then you don't get confused. We talk about cross-reactivity in antibodies, but we talk of cross-inhibition in T cells. So the cross-inhibition means, and this is a good table in your book, that we already know the most important, we call it signature cytokine. So remember the word signature cytokine. So Th1 of all the cells will produce gamma interferon. Th2 will produce IL-4. I just told you the Tregs is a typo. It's not ref, it's a reg. TGF beta. Okay? Now what happens is, since if there's an inflammation, body deals with adaptive immune response in a very specific way. It deals with bacteria totally different. So the point is, for example, if I was to ask you, and I'm going to ask you right now, can you give antibiotics in viral infections? Why? Virus is not bacteria, but they may use the same killing mechanism. Well, you are right, but the thing is, you have to understand the mechanism of action over here. The reason is, not only you cannot give antibiotics, because antibiotics have to be very specific as well, because it's a specific immune response. So no one antibiotic is going to be useful for all infections even. They're gram-positive, gram-negative, so on and so forth. But since the body mounts a different immune response, we want to be very specific. That's why, and I'm pretty sure I'll teach you that, and that's going to be a part of the gram staining lab as well, antibacterial agents, that most of the antibiotics are going to become obsolete. Most of the antibiotics are going to be obsolete because we are developing resistance to them. We're already overusing them. You know where the maximum number of antibiotics are used? In US, for example, where do they use the maximum number of antibiotics? Texas. Oh, you're talking about state, Texas. Why are you from Texas? Okay, cattle. Because they give antibiotics to cattle in farming, because they would want you to become sick because you have an insurance. So cattle don't have an insurance. So they want to make sure they don't become sick, because if they become sick, there's going to be a problem there, right? So they give, majority of the antibiotics used are given over here. And you say, I don't take antibiotics, but indirectly you do. You see the point? So anyway, if it becomes obsolete, what are the new agents? That's why we call antibacterial agents. And the trend is that they're going to be cytokines. So tomorrow we'll talk about what's going to be the new drugs for antibacterial agents, which are cytokines. Now what happens is, and very interestingly, this is nothing that we made it, the body makes it already. We're just learning from the immune system, how do you deal with that? So we want to help you out and compensate you, or help you in terms of producing that thing. Now, one Th1 cytokine signature is produced. It helps Th1 response, but at the same time, it inhibits Th2 response. I mean, just like a if you trip towards one side, the other side is going to go up. It's all a balance there. If you put on a balance, it's automatic. But at the same time, what we are learning is that, I mean, I taught it last year, but I'm learning myself this year, that it wasn't Th2 only, it's also Th17. So that's like a new development. That's why we have Th17. Also, because Th17 produce a cytokine which is IL-17. Okay. Now, when Th2 produces IL-4, it's other way around, but it also inhibits Th17. So what you will see over here, that both of them are enemies of each other, but they have a common enemy, that's Th17. So then there is a problem over here. That's what we have to have a very precise balance. That's why we say orchestrated immune response. Even all those hundreds of violin player or piano player mess up one tone is going to mess up the whole orchestra. Okay? And then when the Tregs produce TGF-beta, and I just told you is a regulator, but I also told you we used to call it suppressor, but still the function is suppression. It is suppressing all those responses. That makes sense? So this kind of an activity initiated by CD4 T cell we call cross-inhibition. Okay? So if I was to tell you allergy is a Th2 response, so this means if you want to control allergy, right? So you would rather give other drug that inhibit that response. So that will be a new drug for you because you want to control allergies. Also, when you have one particular response, for example, parasitic infection, you will also see that you develop allergies with that because the way immune system deals with parasite is more or less the same as we use for allergies. That makes sense? So this is a concept in terms of cross-inhibition. And while I was running through this and I looked at the guys and say, hey, I thought to myself, let me finish it, but it was all blank faces. But this is the most important concept in terms of use of vaccine. So if you understand the use of vaccine, because we had the other day one of the lectures and they were talking about if the job of pharmacists was just to dispense the medicine, then they've already have got robotics working in California. They can do a better job. But robotics would not think. Robotics will just do what they need to do in terms of what is fed to them. Okay? They would not do research and development because there's no mind there. There's no critical thinking. So in order for us to understand, for example, why do we want to make vaccines? What are the advantages? Because the only success story in the medicine is vaccines. So we've been able to control the infectious diseases by vaccine. And tomorrow, if there is a threat for the flu, you need to understand, not only give, because if it's only to give an injection, I don't think that you need an MD or PharmD. Anybody can give an injection. It's not a big deal. It's all written over there. Okay. So that's what happened when the virus gets infected into the system. You see the importance of viruses? I don't want to skip this version, probably. That's what I would do. Okay. It always happens. Whenever I talk bad about viruses, my computer shut down because of viruses. So probably, I don't know, they listen. Viruses are smart. So anyway, so you can see the concept about a very specific protein, which is a flu protein. So you get flu, and everybody gets flu, common cold, right? So also keep in mind that the big drug industry is not going to spend money on development drug for malaria, for example, because malaria is not their problem. Very few cases. So they would go where there's a big money, right? So everybody would need a flu shot. If something happens, God forbid, swine flu or bird flu, then the whole nation, so this is called emergency preparedness. The Department of Health wants to make sure the CDC or NIH is ready to vaccinate the whole US population for 300 million people. So guess what? Where's the money then? It's in that industry. They're going to hire people, MDs and PharmDs all day to work on this project because they're not working on a small percentage of a immunodeficiency that happens to 5 kids. Who cares? They would not care. Is the point? So the idea behind is that it influences even immunologists as well because they get funded from the government and governments will say, hey, we're going to give you money, do research on that. That's why you see most development in those areas. So in this case, there's a flu protein coming from a flu virus. And we already know the basic immunology that this is to be seen by a B cell. And B cell is an antigen presenting cell. Okay? But B cell, again, remember the capabilities of a B cell, that one, it has this immunoglobulin attached as a BCR receptor. It is going to phagocytose the whole structure inside by even phagocytosing the antibody as well. So antibody goes, you remember we talked about this antibody receptor capture where it's internalized and recycling of antibodies. There was one slide I showed to you. So B cell has that capability. So the whole flu protein is going to go in. It's an Ig bound antigen. You can also see it's already bound over here. It's going to be metabolized and processed into acid vesicles, and then it always has a MSC class 2, is going to traffic it to the B cell surfaces and present it. Okay. Now, since this is a class 2, CD4 T cell will come, and now that's what they're talking about. This is like a developmental thing, and I'm pretty sure I didn't see it last year, but it's a new book. So what they now see is that like a specific kind of a CD4 T cell, which is flu protein specific. So they look at the whole signaling events. They look at all those enzymes because they want to make drug, and they finally would look at transcription factor. So the transcription factor they look as over here is BCL6. So when BCL6 goes up, one of the protein that it makes is one of the receptor that's put on to the surface of a CD4 T cell, which is a chemokine receptor or chemokine-like receptor, CXCR5. So now they know the whole story. So they pretty much know this is what happens. Okay, so then they will think, hey, we need to develop this particular kind of CD4 T cells. That's why I'm going to give you a shot. So you develop those particular T cells that are required to fight that flu protein because they are flu protein specific, right? Because it's going to see a very particular part of. Okay, now pay attention to the CXCR5 over here. So this is a flu protein specific T cell. Remember we talked about TH1, TH2, and 17. No, they come up with another one because it's a very specific immune response. If there are 101 infections, we have 101 different types of T cell. We don't actually know what is their particular phenotype or what is their genotype per se. So that's what we are struggling, and that's where the developmental part comes. Now, in order for us, for example, this T cell to produce antibodies, let's say we want to produce antibodies against that particular virus, right? So why did B cell present it to a T cell to make antibodies? Why did the P cell internalize that particular flu protein and present it to T cells? Because it's a protein. Protein are all processed by T cell. B cell cannot make antibodies two proteins without the help of a T cell. So that's where, actually that's where the word helper comes from. That was the origin. It needs help. Okay? There are many things, but one of the most important thing is gene, MSC class 2. But it's a protein. Okay? Now this T cell will come back and again go back to the B cell. It's say, hey, we want to interact. So this is what we call T helper cell and B cell cooperation. They need now to cooperate to produce very specific antibodies. Now when they come together, now keep in mind all those cell surface ligands that are going to be interacted, and they are important because each of them, if like 6 turn on and off, they are going to lead to very specific immune response. Okay? Now let's begin from the most important, the TCR, and you can see from here peptide, MSC class 2, and that's fair. So that's pretty much already established. Then we say CD28 is a co-stimulator. It engages with B7, done. We already talked about. Now the other two things that are important, CD40L talks to CD40 and ICOS talk to ICOS ligand. So these are two extra co-stimulatory molecules that need to be engaged. Okay. Now since the idea is that two important things need to happen over here because we need to make very specific antibody. Firstly, we need to make memory cell against that flu protein so that next time when you get infection, you have memory cell, no need to process the whole information again. They give you a short code, direct dial, let it go. Or you need very specific antibodies other than M and D because remember, you need to have very professional antibodies like IgG. For that, you need another mechanism in the B cell, and that is called class switching. That's called class switching, right? The class switching. And the other important thing we want the B cells to do is to produce those specific, if you remember, how would B cell have so many specific antibodies? One of the things I told was mutation, right? And basically, we call it hypermutation, right? And if you go back and read, we also use the word somatic hypermutation. So we have somatic hypermutation over here. We have class switch and over here, and then we have class switch recombination and somatic hyper, and then we will develop very specific memory cells, and we also develop plasma cells that are going to secrete antibodies. Okay? Sorry?
Specific antibodies or specific cells?
T cells.
No, B cells. Like you said, like the cross-linking and the hyper.
No, I didn't talk of cross-linking.
How do you cross-link?
Yeah, they're two different things. Yeah. So what I'm trying to say is that since this B cell needs to make antibodies, it needs CD4 T cell help, and then we need to have specific cells, which are memory B cells in the system, to take care of it next time when you get that infection. At the same time, we have to make antibodies that are going to take care of this infection right away. Does that make sense? Okay. The third important thing over here is that, remember, we talked about cytokines, right? Now, this thing we already know, but one thing that will happen in particular cases where, remember when I said Th1, Th2, and Th17, and Treg, so I gave you 4 signature cytokines. If I tell you that TFH, which is a flu protein specific T cell, so you need to ask me what is the signature cytokine that this particular cell will produce. Okay, so what we found out that it produces 2 important cytokines. It produces gamma interferon, which is a Th1. It produces IL-4, which is Th2, So that will be kind of a hybrid. It's enough to confuse us, but the good thing is it produces another cytokine, which is IL-21. And don't confuse that with IL-2, which is a growth factor for all T cells. So IL-21. So this IL-21 is required for those specific flu protein to proliferate. Because remember, one of the functions of the T cells is that it is going to interact. One cell will interact with one B cell, but that may not be enough signal, so you have to produce tons of T cells so that they can talk to tons of B cells so that you can have tons of antibodies. So you need, number one, this specific cell to survive, and then you need this cell to proliferate. So what it does is, one, it produces IL-12, IL-21. It also starts expressing receptor for IL-21. So that's an autocrine loop. The cell produces a cytokine, and it comes back and acts on the same cell that produced it. It's an autocrine mechanism for cell proliferation. And then you can see from here, they are there. The other important thing that you will see in this particular case is that this specific B cell, which is a flu protein specific, would also have a receptor for both. So it has a receptor for Th1, Th2. It also has a receptor for IL-21. Okay, so this particular figure will give us an idea that what is it that we require to produce very specific antibodies. Okay? When you deal with a flu protein, which is just a protein, we talk about flu because it's common. Okay. The other concept that we've been talking so far is, why do B cell cooperate with T cell? Why do we need that cooperation? I told you, because of the presentation of protein, because of class switching, and because of somatic hypermutation. So that is important. The other concept in immunology that we call linked recognition. There's another concept, linked recognition, and all these definitions are there in the end of the book. Cross-inhibition, cross-reactivity, adjuvant, any term that is there. So because many of the questions will ask you a term, and then you need to know the definition for that. So this is something we call linked recognition. So what is happening in this case is, there is a protein antigen. So there's a protein antigen, and it's got different epitopes. You can see those branches, epitopes. So this epitope, which is A, or this epitope, which is C, comes over here, and you've been, let's say, vaccinated. So you get infected with this particular protein, or let's say it's a virus, or it's a flu. Now you are hit with B and T cell. The very first thing that you are going to be hit with will be those specific memory cell, T and B cell. So T cell will come over here, and they will look if they have this information of this particular protein, and they say, yeah, we remember that we are very specific for this particular epitope A. And what happens is the B cell also come over here. They are looking for another epitope, because there's a big chunk of protein, and they come and attack it. So TCR engages with the TCR receptor, small amino acid, 12 amino acid, and then antibodies also have that specificity in terms of looking at a sequence of amino acids, so they will attach to it. And then they need to talk to each other because they have just met something which is common, because they want to have a robust response. Okay? As I said the other day, the best response for vaccination is T cell mediated. It's going to stay for a longer time. But we do need B cell as well, because we want to fight it with antibodies as well. So what happened is that now, since they picked up this different epitopes, they need to cooperate. And this cooperation is called linked recognition. This cooperation between T helper cells and the B cell is called linked cooperation, and I'm going to give you an example. Where do we need it and why do we need it? Now, we need this help when we make especially conjugate vaccine. So you will see different kind of vaccines. We have a full lecture on vaccines. For example, you remember you've been vaccinated, but if you're asked what kind of vaccine was it that you were vaccinated for, you cannot answer if it's a conjugate or not. But for conjugate vaccines, you can see from here, we do need that cooperation. The reason is, if it is a polysaccharide alone, which is a carbohydrate, we already know carbohydrates are processed by B cells. So B cell will look it up, they will have a thymus independent response, and they're good at making IgM. Do we need IgM? Well, just to fight infection initially, big molecule, complement activation, complement fixation, class switching, everything. But long term, we don't need that. We need a professional antibody, okay? But if I was to give you this polysaccharide as a vaccine, it's just gonna challenge your B cell and you will make IgM. Not a good vaccine. So we want to make a conjugate vaccine. So what we will do is, we will add a carrier protein to this polysaccharide. As simple as that. Because once you add a protein, whether it be small, then the T cell comes into the picture. So we will have T helper cell, and both of them, they're going to cooperate, and then we get IgG, and that's what we need. So that was the importance of the linked recognition in terms of production of conjugate vaccines. Okay. Now, till this slide, we've been talking about CD4. Okay. Now I'm going to switch gear and move on to CD8, which is another type of T cell. Now keep in mind that CD8 cell interact with MSC class 1. Okay, correct? Now, if you pay attention to, let's say we have a virus or virus-derived protein, and we have dendritic cell, which is a professional antigen-presenting cell. It is going to take it in, present it to MSC class 1, and then CD8 T cell will come attached to it, and then they will have an effector response. Again, effector response pretty much the same. Proliferation, differential subset formation, cytokine production. These are the three major ones for the CD8 T cell, which are called killer cells or cytotoxic cells. Now, what normally they need to do for, let's say, proliferation, because if they need to proliferate, they also need a growth factor. And the growth factor for both CD4 and CD8 is IL-2. So one of the most important things that you want to make sure that IL-2 receptor is put on those cells, and then they start secreting IL-2 in response to antigen presentation. This will get them activated. Remember, I said normally IL-2 receptor is present on T cell, but it is lacking one chain. Do you remember what chain was that was lacking? Huh? Alpha chain. There was alpha chain. There was alpha chain. If you go back, there was alpha chain. When we talked about activation of T cell, I said the alpha chain is something that needs to be put on for the proper functioning of receptor. Receptor is there, but it is redundant. I give example for the pro-hormones. They are not ready to go, okay? And so you can see from here, that's one typical response. The other thing is that this time you get an immature dendritic cell who has not seen any antigen. So it's going to take that in, and this immature dendritic cell can break it apart into different parts. So this cell has capability to present it either in MSC class 1 or MSC class 2. If it presents viral peptide in MSC class 2, the CD4 T cell will come into play, right? And they will make IL-2. Okay? If it presents to MSC class 1, CD8s come over here, right? And then again, they will also get activated. But the good thing is that now in this particular case, the IL-2 is not coming from this cell alone, it's also coming from CD4 T cell. Okay? Now the other difference, as I said earlier, because nothing stops a particular antigen presenting cell to go and see that virus. Anybody can see the virus. So this is a immature dendritic cell, right? Because it hasn't seen it before. But this one is a mature dendritic cell. And as you can see from here, so this may have a prior information of this particular virus. That's why it's mature. The difference between mature and immature is that this produces another cytokine, which is IL-12. And you see we don't have IL-12 over here, and we have IL-12, which is also an activating factor for development of CD8 T cell. So the whole story is that you can have CD8 participation of factor immune response by viruses, depending upon how the viral proteins are presented. If they are presented in context of MSC class 1, CD8 alone will proliferate, right? If they see in the context with CD4, they get an extra kick or production of more IL-2, and you may have an enhanced response in terms of a CD8 effector cells, and both of them are cytotoxic. Okay, now let's see what does a cytotoxic T cell do. Okay, so this is a cytotoxic T cell, right? And then again, in the previous system, you make these cytotoxic T cells. Now, cytotoxic T cells have many things in common. For example, they have an ability that they contain specific granules which are like ammunition for them to kill, number one. Number two is that they have an ability to, if you remember pathophysiology, I said the death tag. So they can come and put a death tag and handle that kind of tag to kill the cell. So you don't want too much of CD8 cells circulating, because if you've got too much CD8 cell circulating, they have an ability to kill. They are cytotoxic. Okay? So viruses may have more ability to cause tissue damage as compared to bacteria because of the way the immune system deals with them. Now what happens is, let's see one particular cell. So you get infected and you have a cold sore, for example, cold sore of the lip, and you will see pain there. You see a lot of different kind of pathology over there, tissue damage. Tissue damage causes pain. So what's happening is, let's take that buccal or cell of your lip and look at it, because it's been infected by a virus. Okay, so it is a virus-infected target, because some of the viruses want to go and hit the liver, some of them want to go hit the skin, some want to go for mucosa, some go for the cervix. They're very specific affinity for different viruses. Some go for respiratory tract, some go for the brain. We'll talk about that in detail, and that's going to be dependent upon the adhesion receptors that they have. Now, regardless, if there is a virus-infected target, right, and you can see this will be broken apart and presented, because it's a nucleated cell, so it's going to present it in context with MSC class 1. That's why you see only one chain over here instead of two. And I also told you one of the most important molecule that is a part of MSC class 1, and this is going to be second time or third time that I'm asking you this question. What is the name of that? Beta-2 microglobulin. Beta-2 microglobulin, and you can see this tiny thing over here, beta-2 microglobulin. that was not the case with MSc class 2. Now, once your cells get infected, the CD8 T cell will engage with that, right? We have a TCR receptor and we have CD8 co-receptor. And what will happen after that is that they will induce pore. So they kind of release a protein we call perforin. It's like a protein which pokes hole into the cell, perforate the cell. We call it perforin. Okay, so you can see perforin. So perforin is going to create the perforin pore. The reason is because it's going to send all those killer granules into the cell to kill it. So one of the effector mechanism for CD8 T cells. The other effector mechanism is that it carries the license to kill, but it has to see the death tag. And so that death tag will come with this interaction, and FAS will be put on. So FAS will tie up with FAS ligand, and by the end of the day, the virus-infected target will die of apoptosis, death of infected target cell. That's how viruses kill. So this tissue could be anything, right? It could be liver, it could be skin, you name it. But these are not the only mechanisms. But I'm giving you just two important mechanisms. When we talk about CD4, CD4 was more of helping, producing cytokines, producing antibodies, more of that nature. But when it comes to CD8, we don't want to wait. CD8 will go for the kill. That's why it's too much killing, too much problem with viral infection. So if you give antibacterial, this is going to help. So we would rather to treat viral infections, so we have to deal with all these molecules over here. Because we need to get rid of this virus, but we don't want the tissues to die. So that's a challenge for viral infections. Okay? So I'll stop here.